Good afternoon, everyone. My name is Dr. Allison Murphy. I'm the immediate past president of the Greater New York region of ACSM. I'm also an assistant professor of exercise science at Mercy College in Dobbs Ferry, New York. And I'm here today to introduce our next speaker. Dr. Andreas Stamatis is an associate professor, the undergraduate program coordinator, and the lab director in the Department of Exercise and Nutrition Sciences at the State University of New York in Plattsburgh. Dr. Stamatis is best known for his research in mental toughness in sport. He's authored and co-authored many peer-reviewed articles, and he's given talks in several countries on this topic. Besides academia, Dr. Stamatis has worked many years as a strength and conditioning coach in Europe and in the US. He is originally from Greece, where he owns and runs a variety of health and fitness businesses. Among other professional organizations, Dr. Stamatis is certified through the ACSM as a certified exercise physiologist. He is also an active member of the Greater New York chapter, where he serves as the member at large of the executive committee. He's also a member of the research committee. He's also on the certification board of the Collegiate Strength and Conditioning Coaches Association and the section editor for psychology and behavior at the International Journal of Exercise Science. Dr. Stamatis speaks Greek, English, and French. He's honored to give the Student Spotlight Lecture of the Fall 2021 Greater New York ACSM Conference. While trying to cover the basics of this new, exciting world that brings together sports and neuroscience, he hopes this presentation will challenge the attending students to get involved and help evolve this new discipline. Dr. Stamatis.
I want you to take a look at the incredible network of blood vessels that exists on the surface of the brain. So you're looking at the right hemisphere of the human brain from a male. Specifically, this is only the cerebrum. We're missing the cerebellum and brainstem down here. But these blood vessels, they just glide over the surface of the brain tissue itself, and they're embedded in the meninges. You can see this tissue that's on top of the surface of the brain, and the blood vessels are inside of it. And then what will happen is these blood vessels will progressively get smaller and smaller, and that's when they will go and penetrate deeper down into the brain tissue itself.
All right. Good afternoon, everybody. My name is Andreas. I'm a professor at the Department of Exercise and Nutrition Sciences at the State University of New York in Plattsburgh. And today I'm very, very excited to talk about that 3-pound mass of fatty, soft tissue that you just saw, and which controls almost every function of the human body. So I use this popular TikTok video to just catch your attention. But in general today, since this is the Student Spotlight Lecture, I'm not going to talk about my line of research or somebody else's line of research or about a very specific topic. We're going to cover a lot of topics, but not in depth. And my goal is, again, and as you can see in the handout that you probably have in your hands right now, is to give you a little bit of a basic knowledge so you understand better what you already saw during this conference, what you're going to see after my lecture, but in general, and most importantly, hopefully, to make you realize how important is this new subdiscipline. And again, hopefully at the very end, your brain wants to learn more about its function. All right. So let's start here. So yeah, we have a lot of big words today, and I'm going to talk about some terms, but we're going to take it slow and cover everything. Do not worry. The most basic thing and the first thing we need to cover is that we already know there is a bi-directional relationship between the environment and our body. So the environment is sending some information towards our body, for example, the light, sound, the temperature, and then our body receives that information and acts or reacts, and that affects the environment. Now, take that and put it in a sporting environment, and that's where sport neuroscience is born. Let me give you an example. So here we have an athlete, we have a runner running outside, we have the environment, we have the sun, we have the ground, we have the wind, which affect the athlete. You see the vasodilation, for example, but also the athlete, by being in the environment and reacting to that, for example, the evaporation, it will affect the environment back. All right. So the most basic point at this point, before we even start, is that sport neuroscience is located where sports psychology and applied neuroscience meet. Okay. So what are we covering today? We're going to cover 4 basic things. I debated a lot, but I've been gathering all this information for months, and we're going to cover these 4 main things. First of all, the perception-action relationship. Then we're going to talk about all these methods we use in neuroscience, and I have that neuroimaging because not all of them have to do with imaging, but I put them all together. And then we're going to talk about biofeedback and neurofeedback, and we're going to end it up with a specific type of biofeedback, which has been very hot lately, and I want to talk about it, heart rate variability, or HRV. What's my objective? My main objective is to create the next sport neuroscientist. Hopefully you get hooked and you like it. I talk with students all the time, and the majority of them doesn't really know where I want to go upon graduation. So today I'm going to offer you maybe new ideas, and who knows, maybe something clicks, right? Now, all jokes aside, I'm going to cover what it is, how are we practicing it, what do we know already, and which are the things that we haven't investigated, which, by the way, are more than the ones we have investigated. All right, let's start, because we have a lot of information, and I don't want to use the whole hour. I want to leave some time for questions. So first of all, let's talk about the neural pathways. We're going to start with the functional organization, and we're going to go clockwise. All right, so we already talked about the bidirectionality between the mind and the body. Okay, so you're there, you see me, you hear me, and your brain is receiving all that information, for example, and puts it all together, and you experience this as one experience. So there are different sensory modalities that are connected in your brain, and your brain is analyzing all that information, comes out with that one experience. What did we find? We found that that connection between those different modalities can be trained. So in a way, we can change your perception of the reality. And if we can change your perception of the reality in the sporting environment, that will most likely change the way you play. And then you can change the environment. So finding that this is something trainable is very, very important to understand from the very, very beginning. And it's kind of the cornerstone of all this. All right. So an example of how you can train how you're perceiving your environment, or as we call it, the peripersonal space, is selective attention. There are things that you can be trained to look at and things that you can be trained to not look at. And if you perceive these differently, of course, as you understand, that may alter your performance. Another thing that is very interesting, and I want to talk about today, that you may see if you go into this field, is mirror neurons. Okay, so here, when you do something, we do know that specific neurons are going to fire. But also when you're watching something, whether that's somebody else live or even yourself via video, mirror neurons are firing similarly. So that is, both of them are very important, because we are trying to teach your brain to recognize specific patterns. And if you recognize those specific patterns, then you can anticipate something. Is that important? Well, think about any interactive sport, such as boxing. What if I can anticipate the opponent's next move? Is that important? Of course it's important, right? So we do know one last thing about this, that expert athletes have their cues, they can identify those patterns, but we also know that they're expert watchers, such as coaches, scouters, or even journalists. So these people also, just from the mirror neurons, they can identify patterns better than others. Let me give you an example. So no matter what's your favorite baseball team, you cannot tell me that me and Ron Washington are going to watch the same baseball game and we are going to understand the same thing. That we're going to watch the same game and we're going to have the same experience. We're going to watch the same players, the game, and we're going to recognize the same patterns, right? And that's why he's an expert and definitely I'm not, right? Okay, so that recognition of the same patterns can help with anticipating movements. And of course, if you can anticipate, you can predict. And that is really, really important because now what we're figuring out through sports neuroscience is that that's an ability. And if that's an ability and you can train it, we can change the whole game, right? There's a paradigm we're using here and I wanted to show you in case you come up to that at some point in your research. There's a temporal fluid paradigm. And
it looks like if there was
as you can
something
see
happening
here,
out there somewhere.
As you can see here,
Looks like it's getting ready.
I'm listening to other people talking in the Zoom. As you can see here, we can have the whole movement in pictures, and then we can take out some parts of the movement. And what we find out is the experts, if you're just watching B, the experts by the placement of the racket, the body placement where the ball is, they can predict where the ball would go. And of course, that's very, very important. All right, so what are those neurocognitive mechanisms? We're trying to figure out why this is happening, right? That underlie experts' superior abilities in, as we just saw, in perceiving, understanding, anticipating, and predicting the sport-specific actions. We have two theories. One is memory, and memory has to do with the perceptual experience. And we already heard about it when it comes to kids in the presentation before me. And then we also have heightened perceptual sensitivity, and this has to do with motor expertise. So perceptual experience versus motor expertise. As you can see on the side, I have a side note about memory, and I want to go there and talk a little bit about that. All right, so stage of learning. We're trying to understand stage of learning, and a way for us humans to understand it, we group it into two stages, and also it's easy to teach it. We have this early stage where we have more cognitive load, less attention in other aspects of the game. And then you move later on to the automatic stage, hopefully, where you have less cognitive load and more attention in other aspects of the game. We have a lot of research with quarterbacks, for example, in the automatic stage. And of course, the goal of every coach is to move their athletes from the early stage to the automatic stage, correct? Now, I'm gonna throw in the hippocampus, and Dr. Suzuki did too. So the hippocampus, it's a structure situated in the MTL, the medial temporal lobe, so deep in the brain, and plays a key role in organizing input to link memories by their contextual representation, right? So I'm gonna go back a little bit and talk about the two processing streams of information. So we have the dorsal pathway that shows us the way. So if I go back to the example of the quarterback, how is the configuration of the line of scrimmage in front of me? That's where I get that information, the dorsal pathway. And also I have the ventral pathway stream, that I'm getting what kind of event or object is in front of me. So information from both those streams converge at the hippocampus. Now let's go back to the stage of learning. In the very beginning, in the early stage, we don't have a lot of memories, right? In that stage, the brain requires more attentional resources. The lack of context in the early stages leaves little work for the hippocampus. But as we practice and practice and practice, the role of the hippocampus become increasingly important, to the point where control processes are no longer needed and the brain can rely on the object event recognition system. Is that important? Well, through neuroscience, we can track where you are in terms of stages of learning. There's a training metric called anterior posterior. So in the early stage, you are anteriorly more active in your brain. And then as you move to automatic, you are more posteriorly activated. So if we can track that, if the coach knows that, they can customize trainings differently. So it's very, very practical, because if you put stress or in the very early stage, you'll have more errors and less reaction time. So if you know where they are, you can customize the practices accordingly. One last thing, let's talk about sleep. All right. Well, I found this picture. Let's talk about consolidating memory with a good night's sleep. So we have REM, and we also have 4 non-REM stages. So in stage 1, it's light sleep. You can wake up very easily. In stage 2, eye movement stops, body temperature begins to drop. In stage 3, it's called deep sleep, slowing heart rate, further body temperature, and we have the first presence of delta waves. Stage 4 is called slow wave sleep, and we have exclusively delta waves. And after that, we go to REM. Who cares? Well, we should care, because REM is associated with emotional integration of memories and creativity in problem solving. I want you to look at the last hours of sleep, 5, 6, 7, and later. As you can see, there's not a linear system. It's not you go 1, 2, 3, 4, and then you go REM and you stay there. We go back and forth. But as we're moving towards the 5, 6, 7, 8 hour of sleep, we get more REM. So if we sleep 6 hours and we lose those last 2, we may also lose as much as 40 to 60% of the time spent in REM, which means 40 to 60% of emotional integration of memories and creativity in problem solving. And of course, is creativity in problem solving important when you are a quarterback? Definitely, right? I just wanted to point that out. One last thing, there is honest, and we call them bluffing kinematics. So what if you fake a movement? Can you see that coming? That is also interesting in terms of neuroscience, because that's called deception. And we've been talking about that embodied representation and how experts have it and they can predict. And now what they need to do is inhibit that embodied representation. So overall, yes, success in sports has to do with perceptual abilities. Yes, it has to do with motor abilities, but also it has to do with cognitive skills. All right. Neuroplasticity. Is neuroorganization a thing? Yes, it is. We thought it wasn't, and even Dr. Suzuki talked about it. Now we're talking about the plasticity of the brain and having several maps, not just one. But it takes time. Is as you train at the gym or you're starting a new diet, it takes time, right? Which one is better, imagery or action observation? So doing imagery on yourself or observing something? I don't know. It depends. Maybe imagery works for you and it's fine. However, what we do know for sure is that action observation is better in terms of time and force, because you can see that in the video or live when you're watching somebody. So you can see the how, which is something you cannot with imagery, and you have to imagine it, which means that you have more mental fatigue. The last thing, and whenever I bring it up in class, students get excited. There is a thing called neurodoping. So we can apply a current to stimulate the cortex while you're learning something. It's more about excitability. There's no pain. It doesn't induce any action potentials. There are two techniques, TDCS and TACS. It's about the TACS has a pattern. That's the only difference. We're not going to talk about it more. But the fun fact is that if you stimulate the cortex while you're learning, that can improve your motor learning. So there is such thing out there. All right. Let's move on. Let's go to imaging, and we'll spend some time here, and we'll talk about the different methods. All right. I'm gonna take you over a scenario. All right. I want you to think about it. You are standing outside the edge of a sideline. You grasp the basketball, as this guy in yellow, between your two palms. You quickly scan the field to get a sense of where your teammates are, your opponents are, and where everybody is in reference to the net. So in that way, you're sending visual information from your retina to your lateral geniculate nucleus to your primal visual cortex in your occipital lobe. The ref's whistle sounds, and it's time to start the play. So you slam your hands against the ball, notifying everybody that it's time to go. So the neurons in your motor cortex communicate with the motor neurons in your spinal cord via the corticospinal tract. Now you see a teammate cut towards the net. You can identify that by the yellow jersey. So the yellow jersey information is sent to the inferior cortex in the temporal lobe via the ventral stream we just talked, while your teammate's position is tracked and sent to the superior cortex in the parietal lobe via the dorsal stream. At the same time, you see another opponent, so your attention shifts. He has escaped the defender and he's just outside the three-point line. You think now, I have two options. Option one, I can send the ball to teammate one. He's right under the net. He's probably gonna get it in. He's been doing it all night long. Now, option two, I can send the ball to teammate two. He's further away the net, but the shot is wide open. So mediated by your prefrontal cortex, you only have seconds to weigh your options. And as the seconds count down, because the whistle has sounded, your medial frontal cortex evaluates your options, and you, for some reason, decide that option two is the preferred option, no defense, wide open shot. Your prefrontal cortex sends the instruction to your motor regions, including your primary motor cortex. Your neurons send information to your motor neurons in your spinal cord via your corticospinal tract. So now your muscles contract, and finally, you send the ball to teammate two, hoping for the 3 points. Complex process, isn't it? And we don't even think about it sometimes. Here, as you can see on the left side, we're trying to teach that. And on the right side, you see a photo from an Oregon game, basketball game, where it's from analyzing games, all right? We're trying to understand what happened. So the complexity of the human brain is astounding. And I'm not gonna go in a lot of detail. It can be divided into various hierarchical regions. The hindbrain, which controls essential physiological functions, such as breathing. And then we have the forebrain, which contributes to higher order function, such as abstract thinking. The brain also, as you may know, has gray and white matter. Gray matter consists mostly of cell bodies and dendrites. White matter mostly consists of myelinated axons. So together, they comprise a series of brain networks that are interconnected and contribute to a healthy brain function. How do we understand all that? Are the brains of the athletes bigger? Are they different in some kind of microstructural level? What is going on? Well, this is why we have all these tools. And I have 7 tools, and I've grouped them in 2 main teams. MRI, DTI, fMRI, and FNIRS are what we use it for neuroimaging, because they're very good at detecting where we have some kind of activity. The other 3 that you see at the bottom, grouped together, they can't really help us with neuroimaging. But I put them in there because they're very popular, so I want to talk about them. 3 out of all these 7, fMRI, FNIRS, and EEG are the most popular. So I'm going to go a little bit in depth about that. So in case you see something interesting, you go for that. All right. This is an MRI, magnetic resonance imaging. What is it? It's based on hydrogen nuclide. Okay. We apply a signal. I'm not going to go into a lot of detail. We have 2 types of relaxation. We have T1 and T2. And because different types of tissue have different types of relaxation rates, we can have these nice images. What do we know about MRI in sports? We do know that in athletes, we have larger gray matter. All right, DTI, diffuser tensor imaging. It looks a little bit psychedelic, more colorful for sure. What it is, it's an advanced MRI. It's based on water diffusion. And we know from DTI that there are beneficial effects of exercise in old age. All right, now we have fMRI. When we talk about fMRI, you need to remember BOLD, B-O-L-D, blood oxygen level dependent. So it can distinguish between deoxygenated, which is paramagnetic, and oxygenated, which is diamagnetic blood. And what do we know? That mainly experts have functional changes thanks to athletic activity. and we're going to talk about that a little bit more as we go. FNIRS, functional near infrared spectroscopy. All right, I told you we're going to talk about some big words today. It's very, very popular, and it can distinguish between oxygenated and deoxygenated hemoglobin, water, and lipids. Okay, and we're going to talk about it in a second. All right, let's go to the three ones that we can't really use for imaging because we cannot really locate where those signals are coming from. All right, MEG, magnetoencephalography. What it does is it just measures magnetic field that is generated by neuronal activity. It's a direct measure of the brain function, and it has a very high temporal resolution. So in terms of time, it's great. In terms of space, not great at all. Another disadvantage, it has very small signals, so we need a lot of shielding to eliminate interference. TMS, this is really important and really different. Now, what are we doing is we induce a current. You see it? So we're hitting the brain with a current. We have electromagnetic induction, and what we're trying to do is temporarily, just temporarily, right? Excite or most often inhibit specific brain regions. So we hit the brain, and then we try to see what happens. It's the only out of all these methods that you kind of can infer causality. The other ones are just observational. And that's why most of this research we're going to talk about today and we have out there is just observational, correlational, okay? And the last one, the EEG, Dr. Ryan talked about it a little bit. So you see how it looks like. We can record activity based on frequency. We have alpha, beta, theta, gamma, what else? Delta, all the Greek letters over there. And we can analyze the information based on that frequency or time, the ERPs that Dr. Ryan talked before me. All right, let's go a little bit more into detail for fMRI. All right, as you can see, it's a huge, it's big. It's not portable at all. What can we talk about fMRI? And Dr. Suzuki mentioned it a little bit. Yes, we know from MRI, fMRI that we can have adaptive changes. And what is very important here is that even adult CNS can change. That's great. In terms of anticipation, we already talked about mirror neurons, and we can see that with fMRI. In terms of prediction, we know that different levels of expertise anticipate different tasks, right? In terms of motor preparation, we know that experts have a more efficient organization of neural resources. And there is a lot of research in dance when it comes to motor imagery. I want to talk about some research that has to do with volleyball because it's a little bit applicable to most of us in here. I don't know if you've ever done imagery. You can think about, positive and negative commands. So do something or don't do something. What we do know is that when you include positive commands, you have a shorter reaction time. So in terms of coaching, if you're prescribing imagery, it looks like the positive commands are better in terms of reaction time. Now, emotional states. That's another thing that has a lot of practicality. Visual and auditory stimuli, fMRI says that, can manipulate our emotions, which can affect our motor performance. So when the time is right, during practice, we need to include visual and auditory stimuli. We need to make it as close to game conditions as possible, because again, fMRI shows very clearly that visual and auditory stimuli affects our emotions, which affect our performance. All right, FNIRS. Hang in there. I know it's kind of a lot, but I wanted to introduce you to more things. The basic idea behind it is that when we have some activity in the nerves of the brain, there's going to be some blood flow. So if we can detect the blood flow, we can indirectly talk about the neural activity. That's the basic idea, right? We talked about the oxygenated and deoxygenated hemoglobin. That's the chromophores we're looking for. And you see those nice pictures that have those nice colors at the bottom right. So we have blue, which is the deoxygenated, and red, that is oxygenated. It's not a binary system. It's a continuum, right? It is very, very popular. Why? Why it is portable, it's non-invasive, it doesn't cost too much, and you can collect that information in real time, which brings me to my next point, which is virtual reality. I don't know if you have seen all these videos with, for example, quarterbacks going over the games, learning their games using virtual reality. So we are avoiding all these concussions Dr. Perrin talked about this morning, but they're still learning. And we can attach FNIRS and get information while they're using virtual reality. The last point, which I personally find interesting, I don't know about you, is that through FNIRS, we can make, we have some evidence to make a statement against that phenomenon of fatigue and that hypothesis that it's only related to muscle structures. So FNIRS shows that when we have oxygenation in one part of the brain, we may have a relevant reduction of oxygenation in ipsilateral or contralateral parts. That's important. On top of that, we know that because of intense, exhaustive physical exercise, we may also have notable decrease in oxygenation. So again, this evidence makes us see that this phenomenon of fatigue is just not about muscles. It includes the nervous system and it includes the brains. All right, one more thing, EEG. Okay, this is in our lab. I don't know if Kyle is there, but if Kyle is there, he better raise his hand so everybody can see him. Kyle was there 2 years ago presenting EEG data. All right, so what do we have here? EEG records postsynaptic potentials, not action potentials. That's one. Dr. Perrin talked about concussions a lot. What we do know is that before I even go there, we said that we can analyze data based on frequency, alpha, beta, and so on and so forth, or time and ERPs, okay? Okay. Dr. Perrin talked a lot about concussions. And what we do know is that they manifest in various ways. They manifest over time, so you can have differences, let's say in the frequency, right after it happens, the event, but also years after, right? And also, what kind of difference do we see? We definitely, there's some kind of consensus. We see a decrease in alpha band, and we see an increase in that theta to alpha ratio. One more thing I want to talk about here, you may see it if you go into that research, is the P300, which is a relatively large position deflection, which occurs approximately 300 or later milliseconds after the presentation of a stimulus. Why do we care about that? It is thought to mark the allocation of attention resources. So people who have concussions have smaller and later deflections, which means smaller and later allocation of attention resources. All right, let's move on. We have a lot of research with EEG and that pre-performance period, which are those seconds before you actually perform. A lot of research in shooting and in close sports for obvious reasons. It's easy to do it there. And we have found, I'm just going to give you correlations. We don't really know why, right? I told you that, that alpha increase. We think it may be because you are in automaticity, you have less analytical thought and visual attention, maybe, right? Can we differentiate good versus bad performance through EEG? Yes, we can see that the beta is lower, and maybe because you're less engaged. Again, we don't know. It's all correlational. Can it differentiate experts against novices? Yes, experts have theta, which is the working memory, and alpha, which is the motor preparation higher. There's also one thing called EEG coherence. So there's lower consistency in the different parts of the brain. And actually, Kyle presented about that. We assume that as you're becoming an expert, there's some kind of hemispheric specialization. Can it differentiate between athletes and non-athletes? We know that in competitive athletes, we have higher theta and alpha. Can it differentiate between disabled versus non-disabled? We don't have enough data. Maybe that's something that you would like to get into. What do we know about learning? Can it help with activity? Yes, we don't have a ton of data, but we do know that learning results in increased sport-related alpha activity. And the last thing, what happens with the social kind of manipulations? We do know that pressure, stimulation from the crowd or the coach or yourself, and difficulty level will have an effect on stress. And stress will have an effect on attention, which will increase EEG activation. So less alpha and theta and more beta. Beta is more about the outwardly focused concentration. All right, one last thing about EEG, in case you go into this. I want to talk about sensor placement. There is an international system, and we'll start from the bottom right. We have the nation, so that's how you put the cap on. You see here, Kyle is practicing, and we're putting the cap on him to test it. So how do we put the cap? We have a region that's called nasien at the front, and we have the inion at the back, and also have two, the left and the right preauricular points. Now, you see a lot of letters and a lot of numbers. F is for frontal, P is for temporal, C is for central, P is for parietal, O is for occipital, but also you see mixed labeling. The numbers now, even are the right hemisphere, odd are the left hemisphere, Z are on the midline. Okay, so if I say the electrode is at F3, you already know because you know the system. If I say FC4, again, you know. Does that make sense? All right. And we also put some electrodes, I would call them reference, usually one at the earlobe and one ground or two ground electrodes for safety. Thank you for your patience in going over all these methods. One last thing, actually. I put here, I made a table from those three most popular ones. So if you're doing research and you're looking for being very specific in terms of space, where the signals are coming from, go for fMRI or FNIRS. If you're going for time, the fastest is EEG. Portability-wise, FNIRS is great. There are portable EEGs, but there's a lot of noise. So the quality of the data that you collect are not very well. They're like Mobita is a good one, but not that good. So that's why I put a question mark. Cost-wise, FNIRS is affordable. EEG is affordable as well. Causality, none of them. You can use that data to infer causality. It's just observational. Okay. Hopefully, this is helpful for you to understand a lot if you read any research that have to do with sport neuroscience, especially the methods part. All right. Time-wise, okay, we're good. All right. Feedback. Biofeedback versus neurofeedback. So both techniques, I'm going to talk about similarities, differences, the efficacy, and some other stuff. So both techniques have the roots deep in the operant conditioning paradigm. The biofeedback is about biosignals, such as heart rate or blood pressure. And of course, the neurofeedback is about brainwaves. Now, what is the operant conditioning paradigm? We're talking about reinforcements and punishments, right? So those are used to implicitly promote learning and eventually to save behavior. And then apply these principles and techniques to help people build a strong body awareness and to learn how to influence those responses during actual games. So here we are supporting that physiological status affects mental status, and mental status affects physiological status. It's a more holistic approach, okay? So this is why this is psychophysiology. The very first step is awareness. That goes along with something very popular lately, which is mindfulness. So in the beginning, you have to be aware of your brain waves or your heart rate. Yes, okay? And then after you find that, we identify which are the optimal zones for you. And then the goal of any coach is slowly, you don't need the coach and you can self-regulate. So you take that from the lab to the field, okay? Side note again, most of the research is in the lab. So there's a lot of debate about the ecological validity of all this. But we have some examples, some big examples that work. For example, the Italian national football team, 2006 World Championship, they called neurofeedback training as a secret weapon. So there are teams that are already doing that. Now, is feedback, neurofeedback and biofeedback better than relaxation techniques or self-talk? In my opinion, yes. Why? Because it informs the practicer, we're talking the person who's doing it, how their body reacts to those mental tasks in real time. So you have that feedback, okay? That quantifiable feedback. And of course, we're not talking about any kind of electrical stimulation or anything. When we're talking about reinforcement, maybe when you control it, you listen to your favorite song and you know you did it. And when you don't control it, you hear a beep. Okay, that's what we're talking about. All right, and last thing for today, a very specific type of biofeedback, the infamous HRV, okay? That supports that continuous relationship between mind and body, which is exactly what psychophysiology is about. All right, so I have a lot of things here, so I put them on those sticky notes, and I'm gonna, as I talk, I'm gonna mention them. Let's talk about the baroreceptors, first of all. The baroreceptors, they can have some inhibitory control in the cortex, right? Which can mean less cognitive activity. And we care about that, hold that, okay? We have the sympathetic nerves and we have the parasympathetic nerves. So now you're sitting there, you're all calmed down. Both are active, just that now that you're calm, parasympathetic nerves are more kind of in charge, right? So in general, what happens to our blood pressure or our heart rate in this specific example is the net effect of the parasympathetic to the sympathetic. Now, when the parasympathetic is active, we get that reaction to the heart rate immediately. So it slows down. When sympathetic is active, it may take up to 5 seconds. So from a convenience kind of standpoint, it's easier for us to measure that slowdown of the heart rate, the activation of the parasympathetic. There are two ways, mainly two ways, to talk about HRV. We can talk about frequency. Here you can see very low frequency, high frequency, very high frequency. A classic metric is the low frequency to high frequency ratio. And also there's IBI, which is more interesting for me tonight. And that's what I'm going to talk about, which is the interbeat interval. So from R wave to R wave, that's a heart period. So based on that, we can have HRA, acceleration, or HRD, deceleration. If that interval is going down as a number between the heartbeats, it means that your heart rate is going up. So that's an acceleration. All right, think about it. Now, if that number is going up, it means that those heartbeats are further away. So your heart rate is going down. So we call it heart rate deceleration. Why do we care about that? First of all, I told you that we can actually see the parasympathetic nerves actions or the reaction in the heart rate immediately. So that's convenient, but pay attention. HRD appears automatically when a stimulus is anticipated, those seconds before a stimulus. By stimulus, it can be shooting a free throw, or it can be some kind of reaction, so hitting the baseball. So you're waiting to hit the baseball with the bat. So HRD appears right before that. But, and that's important, it can be disrupted or not manifested in those critical moments, maybe because of stress, maybe because of negative emotions. Why do we care about that? Because when we have greater magnitudes of HRD, we have faster reaction times. Okay, so this is something really important. And actually, with one of our grad students, we submitted a proposal for NCAA to do some research on HRD and baseball. Now, I created a table here to put them all together. So HRD we like and we want to investigate it is a biomarker. I want you to remember that of physiological readiness. And this is pretty much what I just mentioned in a table. All right. Yeah, I know. That was it. All right. Topics that we did not cover. We didn't really go over applications or interventions. I told you that we're going to do mostly theory. There's a lot of research in GAZE, mental training protocols, neurofeedback training. Other people went over the clinical type, like TBIs, concussions. We didn't talk about that. There's a lot in technology. There's a lot in technology. Maybe you are an engineer, or maybe you know an engineer and you have an idea and you create the next best portable mobile EEG? I don't know. Think about it. Okay, you're mostly undergraduates in the audience, so think about it. There's so much to do. And we didn't also talk about certifications. There's a Biofeedback Certification International Alliance. You can go to their website. Biofeedback is a skill, so you need to be certified that you can actually do it. So what did we talk about? We talked about that we can change the perception of our reality, and then that will affect our game and the environment, the sporting environment we're participating. We said that when we're going to look for time, EEG is better. When we want to look for location in the brain, fNIIS and fMRI is better. We talked about feedback, and that the first step is awareness, and then creating those optimal zones, and then self-regulation during those critical points that you can bring your heart rate or your blood pressure or your waves up to the point that is optimal for you to perform. And we gave a specific example through HRT, and we like HRT. We want HRT before those critical moments because it affects positively the reaction time and the task performance. Thank you so much for your patience going through all this. You have the ways to contact me on your handout, but please ask me any questions you may have.
Any questions? Okay. Thank you, Andreas. It doesn't look like there are any questions.
Oh, okay. All right. No problem at all. Then I want to finish by thanking Gali and the rest of the executive committee. This is an amazing conference, and the topic alone is so special. And I want to tell the audience, you're not going to find a lot of conferences talking about sport neuroscience. So this is a very unique experience, and I hope you're enjoying it. I want to thank Tim for the teams and the research part. He organized very, very nicely. And since I talked about the research, I want a special shout out to my students, Janil, Madison, Wills, Dylan, Liam, and Nick. They killed it. They make me really, really proud. I want to finish with one last thing. Let me go this way. And I will show to everybody how much we know. I'm only going to use 10 seconds of this video that shows everybody how much we know about that 3.5 pounds of fat and water that controls everything. Let's see.
Trying to introduce the study of the brain to a bunch of students. I said, if understanding everything you need to know about the brain is a mile, I asked the students, how far had we walked in this mile? I got answers, 3/4 of a mile, half a mile, a quarter of a mile. And I said, I think about 3 inches.
That was it, guys. Thank you very much. It was a pleasure. I really appreciate you. Thank you.